The University of Wisconsin-Madison conducted a landmark 20-year study on calorie restriction using rhesus monkeys, primates that are genetically really close to humans. In this video, I will take you through that experiment and show you exactly how cutting calories affected the way these animals aged. Hi everyone, my name is George. I'm a biologist with 14 years of experience in fundamental research, and for the last 6, I have been focused on human clinical trials. Let's take a look at the calorie restriction through the lens of science. In the late 1980s, researchers at the University of Wisconsin-Madison decided to test a simple idea. If animals eat less, will they age more slowly? More specifically, they wanted to find out if the reducing calorie restriction intake could maintain a higher quality of life and delay the onset of age-related diseases. Things like cancer, heart diseases, kidney issues, and brain atrophy. By that time, it had already been proven that calorie restriction extends the lifespan of yeast, worms, and rodents. However, scientists were still debating whether it would work on long-lived animals. We needed a definitive experiment. Rhesus monkeys were chosen because they are 97.5% genetically identical to humans. As they age, they develop the same diseases we do: diabetes, dementia, and cancer. They are also really similar to us in terms of diet and behavior. A great example is the monkey punch. Anyone who has seen the footage can't help but feel a sense of empathy. The study began in 1989 with 30 males. In the 1994, they added another 30 females and 16 more males. This larger sample size was necessary to track rare diseases and to see if calorie restriction affected males and females differently. The monkeys were between 7 and 14 years old at the start, essentially full-grown adults. Considering rhesus monkeys live about 27 years on average. For the first few months, researchers established a baseline calorie intake for each monkey. Then, they split them into 2 groups matched for weight, sex, and age. The calorie restriction group food intake was reduced by 10% each month until they reached a 30% reduction from their baseline. They were fed on a strict schedule. The control group were allowed to eat as much as they wanted, but only during a 6 to 8 hour window each day. The nutritional quality of the food was identical for both groups, however, the diet was high in refined sugar. And over time, the control monkeys who were free to overeat began to develop obesity. A decade into the experiment, the difference were striking. Just look at these photos. The monkeys on the restricted diet looked significantly younger. They kept their costs, maintained upright posture, and didn't develop the abdominal fat typical of aging primates. Naturally, the caloric restriction group weighted less than the control group. But here's the interesting part. Sarcopenia, the age-related loss of muscle mass, started much later in the caloric restriction group. DEXA analysis revealed that caloric restriction monkeys maintain higher skeletal muscle mass. When normalized for body weights, the upper leg muscle mass of the caloric restriction group was significantly greater than controls, despite the caloric restriction animals being lighter overall. The diet group also stayed more active. These graphs show the time spent in different levels of physical activity. On the left, low intensity, sitting and looking around. On the right, moderate intensity, walking. The more intense the activity, the bigger the gap between the groups. The monkeys on the diet were consistently more active than the controls. The strongest argument of the calorie restriction was the improvement in quality of life, specifically the age at which they developed diseases. In the control groups, signs of type 2 diabetes appeared just 6 months into the experiment. By the time of the final analysis, 16 out of 38 control animals were either diabetic or pre-diabetic. Look at this graph. X-axis is the age of animals. Blue line marks the control group. Red lines reflected the caloric restriction group. Markers on the lines individual diagnoses. In the caloric restriction group, not a single case of impaired glucose metabolism was recorded, even in monkeys that had borderline markers at the start. This suggests that calorie restriction doesn't just prevent decline. It can actually improve metabolic health beyond the starting point. Check out this table. Both groups started with similar insulin sensitivity, and after 8.5 years, sensitivity in the control groups dropped by more than half. which is classic sign of age-related insulin resistance. Meanwhile, in the diet group, sensitivity nearly tripled from the baseline and ended up 5 times higher than that in the control groups. What did all this lead to? Out of the original 76 monkeys, 37% of the control groups died from age-related causes, compared to only 13% in the caloric restriction group. Put another way, at any given point, the control monkeys were 3 times more likely to die from an age-related diseases. When looking at the all-case mortality, which includes non-age-related deaths like accidents or anesthesia complications, the caloric restriction group still appeared to survive better. On the graph, the red line stays above the blue line, meaning more animals remained alive over time. However, mathematically speaking, the difference in all-case mortality wasn't statistically proven. By the way, if you want to see the full study, you can find the link in the description of this video. Just like humans, rhesus monkeys see spike in cancer as they age, the most common being intestinal adenocarcinoma. The incidence of tumors was 50% higher in the control group. The same went for cardiovascular disease. It was 50% lower in the caloric restriction group, though the total number was small. 4 cases in the control versus 2 in caloric restriction group. In humans, heart disease is the #1 age-related cause of death. So this is crucial observation. One of the most modern aspects of the study was the live observation of the brain. This graph shows the amount of gray matter in the singular gyros, an area linked to emotional regulation and stress response. Blue line, caloric restriction group, slopes upward, and green line, control group, slopes downward. This means that while the control monkeys were losing gray matter, the caloric restriction diet at the very least prevents that loss. Similar results were found in the caudate nucleus and the putamen, which helps prevent fine motor skills. As you can see, the average values for the diet group are significantly higher. The takeaway is clear. The animals on the restricted diet lived longer and their brains aged more slowly. When these results were published in the journal Science, it was a total bombshell. Now you can see why. Since then, more studies have been published on both monkeys and humans, and I will be breaking those down in my upcoming videos. So make sure to subscribe so you don't miss them. I've tried to look at these results objectively, highlighting both the breakthroughs and the parts that remain unproven. If you have any questions, leave them in the comments and I respond to everyone. And I also welcome your feedback and critiques. Thanks for watching.